DNA replication. Marc was not able to participate in the recording today.
DNA structure. A DNA molecule consists of two strands connected by hydrogen bonds in a double helix structure. Each strand is made up of nucleotides, which each contain a phosphate group, a sugar group, and a nitrogen base. There are 4 different kinds of nitrogenous bases: adenine, thymine, guanine, and cytosine. The 2 strands are antiparallel, meaning the 5' end of one strand aligns with the 3' end of the other. There are specific points on the strand which are called origins of replication. This is where pre-replication complexes will bind to begin the process of DNA replication.
Step 1. DNA replication begins with the unwinding of the DNA strand, initiated by the enzyme DNA helicase, which uses free energy from ATP hydrolysis to change shape and wedge into the DNA, locally breaking hydrogen bonds between bases on the 2 strands and separating them. The DNA unwinds and replication proceeds in both directions, forming replication forks.
Step 2. Before nucleotides can be added to the divided strands, an enzyme called primase needs to synthesize an RNA primer for DNA replication. The primer creates a template for DNA polymerase to add onto it. When the primer is complete, primase is released and the DNA polymerase binds and synthesizes new DNA.
Step 3. One newly synthesized growing strand, the leading strand, is oriented so that it can grow continuously at its 3' end as the fork opens up. Triggered by RNA primase, the enzyme DNA polymerase sits near the replication fork and adds nucleotides to the leading strand one after another. Polymerase always pairs base adenine with thymine and guanine with cytosine. There are 5 different known types of DNA polymerases found in bacteria and human cells. In E. coli, polymerase 3 is the main replication enzyme, while polymerase 1, 2, 4, and 5 are responsible for error checking and repair. Therefore, during replication, DNA polymerase 3 binds to the strand at the side of the primer and begins adding new base pairs complementary to the strand.
Whereas the DNA polymerase on the leading strand can simply follow the replication fork, on the lagging strand, the enzyme must move away from the fork because DNA polymerase must move in the 5' to 3' direction. Synthesis of the lagging strand requires the synthesis of short, discontinuous chunks of the DNA sequence, known as Okazaki fragments. These fragments are stretches of 100 to 200 nucleotides in humans, 1,000 to 2,000 in bacteria, that are synthesized in the 5' to 3' direction, away from the replication fork. The enzyme DNA ligase then catalyzes the formation of the phosphodiester linkage between the Okazaki fragments, making the lagging strand whole.
Step 5. DNA replication occurs semi-conservatively, meaning that it ends with 2 new strands created from the original strand, each containing 1 old and 1 new strand. With semi-conservative replication, each strand of the parental DNA acts as a template for a new strand, which is added by base pairing.